Glycogenolysis is the breakdown of glycogen to glucose 6-phosphate and glycogen. Glycogen branches are catabolized by the sequential removal of glucose monomers via phosphorolysis, by the enzyme glycogen phosphorylase. Mechanism: the overall reaction for the breakdown of glycogen to glucose 1-phosphate is glycogen, N residues, plus pi glycogen, N1 residues, plus glucose 1-phosphate. Here, glycogen phosphorylase cleaves the bond linking a terminal glucose residue to a glycogen branch by substitution of a phosphoryl group for the I plus or minus 1 of 4 linkage. Glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme phosphoglycoconjugase. Glucose residues are phosphorylized from branches of glycogen until 4 residues before a glucose that is branched with AI plus or minus 1 of 6 linkage. Glycogen debranching enzyme then transfers 3 of the remaining 4 glucose units to the end of another glycogen branch. This exposes the I plus or minus 1 of 6 branching point, which is hydrolyzed by I plus or minus 1 of 6 glucosidase, removing the final glucose residue of the branch as a molecule of glucose and eliminating the branch. This is the only case in which a glycogen metabolite is not glucose 1-phosphate. The glucose is subsequently phosphorylated to glucose 6-phosphate by hexokinase. Function: glycogenolysis takes place in the cells of the muscle and liver tissues in response to hormonal and neural signals. In particular, glycogenolysis plays an important role in the fight-or-flight response and the regulation of glucose levels in the blood. In myocytes, glycogen degradation serves to provide an immediate source of glucose 6-phosphate for glycolysis, to provide energy for muscle contraction. In hepatocytes, the main purpose of the breakdown of glycogen is for the release of glucose into the bloodstream for uptake by other cells. The phosphate group of glucose 6-phosphate is removed by the enzyme glucose 6-phosphatase, which is not present in myocytes, and the free glucose exits the cell via GLUT2-facilitated diffusion channels in the hepatocyte cell membrane. Regulation: glycogenolysis is regulated hormonally in response to blood sugar levels by glucagon and insulin, and stimulated by epinephrine during the fight-or-flight response. In myocytes, glycogen degradation may also be stimulated by neural signals. Clinical significance: parenteral administration of glucagon is a common human medical intervention in diabetic emergencies when sugar cannot be given orally. It can also be administered intramuscularly. Pathology: mutations at the liver enzyme glycogen phosphorylase led to glycogen storage disease 6. Mutations at the muscle enzyme glycogen phosphorylase led to McArdle's disease. Deficiencies at the glycogen debranching enzyme lead to glycogen storage disease 3. See also, glycogenolysis. References. External links: The Chemical Logic of Glycogen Degradation at UFPPT, Glycogenolysis at the U.S. National Library of Medicine. Medical subject headings.